Oh, I hope that works. We'll see. Okay, let's go ahead and get started. Sometimes technology is not my friend. Okay, that'll work. RNA stands for ribonucleic acid. Oh my God. I'm looking to move you. Okay, so RNA stands for ribonucleic acid. This actually comes just like deoxyribonucleic acid. This comes from the sugar that is found in the molecule of RNA. Okay, so type of biological molecule, I'll just kind of move this back and forth. This is a nucleic acid. There's two types of nucleic acids, DNA and RNA. Can we see that okay? Is that good? Okay. We talked about, in chapter 2, 4 different biological molecules. We talked about proteins, carbohydrates, nucleic acids, which we're talking about more now, and lipids as well. Okay? So RNA is one type of nucleic acid. Its structure is it's got some similarities to DNA and some differences. Okay? One major, this guy right here, this represents our DNA or RNA. So label this RNA. And then this guy, of course, right here, this guy is DNA. Okay? The first major difference between the two structure-wise is this guy is a single strand, still twisted, and this guy is a double strand. So under your structure, the first thing we are going to write is we're going to write single-stranded. So one single strand. Our RNA molecule is made up or composed of nucleotides. And if you guys remember, nucleotides are made up of a sugar, a nitrogen base, and a phosphate molecule. So RNA is composed of nucleotides, and those nucleotides have a sugar molecule, a nitrogen base, and a phosphate. Oh shit, I didn't mean to do that. Stupid technology. Okay, so let's look at, I'm going to put NB for nitrogen base. I think you guys can remember what NB means. There's 4 different nitrogen bases in RNA. There is adenine, which you guys are familiar with. There is cytosine, which you're familiar with. There is guanine. There's no thymine. But we have a different base that starts with a U. So U stands for uracil. Okay? So we still have the same base pairing rules. Okay? So let's put base pair rule. Extremely similar. Okay? So we have A is going to match with U. Okay, that's one scenario. C matches with G. We'll just stick with that. Okay. So no T, but we've got U instead. And then the last one, and I am running out of room, is our sugar. Our sugar is not deoxyribose, it is called ribose. Okay, so if you come over here to your picture, these represent our nitrogen bases. And if you look, these two are identical, these two are identical, these two are identical. This is where there's a little bit of a change. You've got this CH2 here, but you don't have one here, okay? So the bases found in RNA, this is cytosine, this is guanine, this is adenine, and this is uracil. And then in DNA, cytosine, guanine, adenine, and thymine. So the big difference is that nitrogen base. Now there's 3 types of RNA that we have to be familiar with. I'm wondering if I need to switch pins to get this all in. Okay, so the first type, number 1, is called mRNA, and that is messenger RNA. So mRNA stands for messenger RNA. His function is to make a copy of DNA and take the information out of the cell. Okay? So make copy of DNA and then take out of nucleus. I said cell. Take out of nucleus. So DNA cannot leave the nucleus unless we're dealing with cell division. And if you guys remember, in prophase 1, the nucleus disintegrates, it disappears, the chromosomes align themselves in the center of the cell. You guys remember all that? Chromosomes are pulled apart, cytokinesis happens, all that good stuff. So that's the only time that chromosomes or the DNA can leave the cell. Okay? It's basically stuck inside of the nucleus, and the only way to get the information out is to have another molecule make a copy of it and then take it out of the nucleus. So that's the job of messenger RNA. Okay? Your second type of RNA is called tRNA, and that is transfer RNA. And his function, well, he kind of hangs out in the cytoplasm. His job is to hang out in the cytoplasm. He goes around, he snags amino acids, and then he brings them into the ribosome so proteins can be made. Okay? So his function is to grab amino acids and bring into ribosome to make proteins. You guys see that okay? You still good? Okay, you have young eyes. Okay, and then our very last RNA is called rRNA, and that is ribosomal. And ribosomal RNA, its main function is to help with the structure and help in making ribosomes. Okay, so function, help to make ribosomes. Ribosomes are important because that's where proteins are actually made. Now, as we move on to protein synthesis and the first step of it, we are going to pay close attention to the first 2 types of RNA. Those are the ones that play a really big part. Okay, so move to your protein synthesis. It's a little bit better, kind of.